This is a lecture on genetic variation in forest trees, and I always start out this lecture with this image, which is not from trees. But what this shows is genetic variation in brassica, in vegetables derived from a single species of plant. So broccoli, Brussels sprouts, kale, cauliflower, kohlrabi, all the same species of vegetable. And to me, this is just a good reminder that there is significant genetic variation in the natural world, even within species. And that can be important to remember, especially when we're dealing with forest trees sometimes, which have enormous ranges. And to remember that both across those large ranges, but even from tree to tree, there can be a lot of variation in genetics there. So genomes are remarkable things, and genetic variation is remarkable. So that's almost always there, although we will talk about some exceptions, some species where there's actually not a lot of genetic variation, but those species are sort of exceptions to that rule. So how does that genetic variation arise? All of the genetic variation that we see arises from mutation and recombination. So mutation is just somewhat random changes that occur due to mistakes in the genome. And sometimes those mistakes, those mutations, end up resulting in altered phenotype that may be beneficial to the organism. And when that's true, then those mutations get retained in the genome and basically multiply through and become more common in the population. Whether that is the case or not depends on the environment. So the role of the environment is it determines whether a mutation is beneficial or not, or whether a non-beneficial mutation gets removed from the population. So natural selection plays out via the effect of the environment on those mutations. Recombination is also important. So when sexual reproduction occurs, recombination happens. So the genes in two parents get mixed up, sometimes in unique ways, and that also then affects the variation that occurs in population and selection that can occur via differences in advantages or disadvantages in any particular environment. So I'm going to play a video that sort of explains or talks about some of those concepts.
Imagine a world where only one population of organisms exists, and every member of that population has the exact same genetic material. Everyone has the same eye color, hair texture,
ear shape, and everyone is left-handed.
Creepy, isn't it? Fortunately, it's also totally impossible. Our world is filled with and shaped by genetic variation. Genetic variation comes about through two processes: mutation and recombination. Mutations are DNA misprints that occur during cell division. Recombination is when alleles get shuffled around during sexual reproduction. Because of these processes, when an individual is born, it will have variations in its genes that were not present in its parents. Some variations help or harm an individual's ability to survive and reproduce. A gene variation in a bird that makes it unable to see color is harmful because it makes it harder for the bird to find food or spot a mate. And a variation that makes it unable to fly could be deadly. Most harmful variations are quickly eliminated from a population. Members that have those variations are less likely to reproduce. Other gene variations are helpful. A plant that's more attractive to pollinators than the ones around it might have a better chance of passing its genes to the next generation. Helpful variations tend to stick around in a population, since the organisms that have them are likely to reproduce. But most gene variations are neither good or bad, just different. Minor changes in ear shape, fur texture, pattern, eye color, and many other visible traits are neutral. Because they don't affect survival or reproduction, they typically stay in a population. That's why many populations have multiple alleles for most genes. Whether a variation is harmful, neutral, or helpful often depends on context. A long neck can be an advantage for animals that eat leaves from tall trees, but if they live in grasslands, not so much. Small, leathery leaves could be helpful in a sunny, dry environment, but in a wet, cloudy place, they might not gather enough sunlight. Because conditions often change, some variations are held in balance. A larger, more muscular body might help an animal escape predators, but because a larger body has greater needs, it could be a liability during a food or water shortage. Bright colors make a fish more visible to mates, but also to predators. The weeding out of harmful variations and accumulation of helpful and neutral variations makes a population more resilient. Populations with little variation are often vulnerable to extinction, and environmental change could be disastrous. Populations with more genetic variation are more flexible and robust. Even if a new virus comes along or the environment changes and most of the population is wiped out, there's a better chance that some members will survive, reproduce, and pass on their alleles. Variety, as they say, is the spice of life. Or variation is the spice of life. Well, actually, variation is essential to life because it makes things more interesting, but it's also necessary. Yeah, okay, you get it. I'm done.
So those are the causes of genetic variation, the mechanisms by which genetic variation arises. Genetic variation can be categorized in a couple of different ways and can be called a couple of different things. When people are talking about genetic variation in trees, sometimes they refer to provenance. Provenance is sometimes used to refer to the geographic origin of seed. So for instance, if seed was produced by a tree that was naturally growing in a particular geographic place, that seed would, let's say it was North Florida. We would say that that seed was from a North Florida provenance. The implication there is that seed from a tree that naturally occurs in a particular environment is therefore fairly well adapted to that environment. Certainly environment is not constant, but at least before climate change, the assumption is that a tree that naturally occurs in a location is fairly well adjusted and adapted to that location. So provenance is a term that you'll often see in the genetics of forest trees. There's a couple of types of genetic variation that are important. There's clinal variation and ecotypic variation. So clinal variation refers to a gradual change in gene frequencies associated with a gradual change in an environmental factor across a species range. So this graph is a nice example of clinal variation. This is the species is Sitka spruce, and they're showing clinal or gradual variation in the date of growth cessation. So this is days since January 1st versus the latitude of that seed source. So the latitude of the provenance for that seed. And these are all grown in one location. So this is a common garden study. In this case, this common garden is in Germany. So this is not a German species, but what it shows is that seed that was taken from a seed source further south grows longer into the year than seed that was taken from a seed source further north. And intermediate seed sources stop growing gradually greater into the year. So that variation is continuous. So this experiment, again, is in Sitka spruce. This is one of my favorite species. This is from coastal Oregon. And Sitka spruce is a coastal species. It grows within a few miles of the coast from, there's another picture of Sitka spruce, but it grows from Northern California along the coast all the way up towards Sitka, Alaska. So peninsular Alaska. And so this study took seed from way north in that range, from way south in that range, and from in between in those range, and planted those seeds in a nursery in Germany and measured when they stopped growing. The important thing about that type of study, a common garden study, is that they're all now, those seed are now all growing in a common environment. So the differences that we see are just due to the genetics of those seeds because the environment is uniform. So that's part of the power of a common garden experiment. But at any rate, this is a nice demonstration of clinal variation in genetics. And in this case, it's due to latitude. Now, latitude is often shown in these types of studies as a source, a geographic source of variation in the environment, but clearly latitude is not an environmental factor. Latitude is obviously a proxy for a lot of other factors that vary as you go north-south. Day length, temperature, sometimes precipitation can vary significantly with latitude. So latitude tends to fold in a lot of other environmental variables. Elevation is another proxy for environmental variation that can be really important. Just like latitude, it's a surrogate for change in environment that occurs with changes in elevation above sea level, often temperature and precipitation. Sometimes when we look at variation in latitude and variation in elevation, those two can substitute for each other. So for instance, there's a rule of thumb that an increase in one location, an increase in 100 meters in elevation, environmentally, at least in terms of temperature, is roughly equivalent to 1 degree of latitude going further north. So going up 100 meters in elevation is roughly equivalent in temperature regime to going 1 degree latitude north. So sometimes when we're thinking about, for instance, equivalence in environment that seed sort of is adapted to, sometimes those elevation latitude trade-offs can help us to, for instance, move seed around on the landscape. Temperature is another important environmental factor. So obviously temperature varies across the continental United States, as you might expect. This is average minimum temperature during the year in the continental United States. And often those minimum temperatures are the most important driver of variation or of adaptation, usually more important than maximum temperature or average temperature in any particular location. So those minimum temperatures can be an important factor. Precipitation can also be a really important factor. And you can see here in the United States, precipitation varies more with longitude than with latitude. So because systems tend to come in off the coast carrying moisture, you tend to see a drop off in precipitation as you move towards the center of the country. And then, so that's pretty clear in the eastern United States. In the western United States, that's more complicated because we have mountain ranges in the west. So we tend to see, for instance, moving towards the central part of the landmass, moving in from the Pacific, we tend to see really sharp increases in precipitation as you move up the west side of mountain ranges, and then a sharp drop off in precipitation as you move into the rain shadow to the east of those mountain ranges. So precipitation varies both with sort of distance from the ocean, but also with geographic features like mountain ranges. So precipitation is a complicated index or environmental factor. The other thing that's also important is keeping in mind that precipitation may not necessarily be the proximal variable that's most important for vegetation. So an example I often like to give, because I grew up in the central part of the country near Oklahoma City, where average precipitation is maybe 32 to 34 inches of precipitation a year. That's basically in the Great Plains. There aren't continuous canopy forests where I grew up in central Oklahoma. But if you go to where I went to graduate school in Seattle, Washington gets about the same amount of precipitation, probably 34 to 36 inches of precipitation a year in Seattle. But there certainly is continuous forest cover in Seattle. They call Seattle the Emerald City because it's covered in trees. So with the same amount of precipitation, there's a big difference in water availability, for instance, to forests in Oklahoma City versus Seattle, because in Seattle, there's a lot less evaporative demand. Summers are cooler and there's not as much evaporation. So it's really the balance between precipitation and evaporation which drive differences in forest cover, for instance, associated with water availability. So that's all in reference to clinal variation. So when we're talking about, variations across the range of a species in the environment and gradual changes in genetics, for instance, across relatively large distances, that's an example of clinal variation. But we also see ecotypic variation, and that's a relatively abrupt change in genetics over a relatively small spatial distance. So you may travel just a few miles in a species range and see a sharp or abrupt change in genotype associated with that. And in that case, there are other factors that are driving that variation. Sometimes those differences can be due to soils. So for instance, a sharp change in soil type, but generally they're associated with historical separations of populations. And I'll give you some examples of ecotypic variation in forest trees that are really important and what historical factors drove those. Another type of genetic variation that occurs is over very short distances. So for instance, within a stand. So if you walked into a stand of trees and measured the genetic variation among a naturally regenerated stand of trees, there would be significant genetic variation among those trees in that population. And that's within stand genetic variation. And that's really genetic variation just due to recombination, or in some cases, mutation. That level of genetic variation is still significant. It's still quite large. And it's really that type of genetic variation that tends to be exploited by tree improvement programs. So tree improvement programs don't necessarily need those large cross-range types of variation associated with clinal or even ecotypic variation. That sort of naturally occurring genetic variation due to recombination within an individual stand can be sufficient to, for instance, select for resistance to disease or variation in growth rate or other traits. So that within stand variation is significant as well.